Good morning, everyone. Great to have you here.
Yeah, so I'll probably be a few people pop in on the next few minutes. I just turned the waiting room off, so people should be able to join. I'm gonna go ahead and pop up a couple polls while we're waiting for a few more people. So if you wouldn't mind filling that out, that'd be great. Just while people are doing that, start with a few introductions. So, hi, I'm Christy Lawton. I'm the Chief Science Officer at Rewire Neuro, and some of you have probably seen me before on a few of these webinars and interacted via email. So today we'll be covering, we'll be focused on our new AutoML custom model training pipeline, which is a humongous improvement over our old model training. And John will be showing you not only how that works and explaining why it's so much better, but we'll also be showing off our new Pipsqueak Pro 2.0, which is a pretty big upgrade to our user interface as well. There's a lot of new features that make it a lot easier to use, a lot more powerful as a system. So, we have Grant Wade here, our lead software developer, who's, been instrumental in making all these changes, who will be here to help answer questions as well. I'll turn this over to John in just a sec. I just wanted to let everybody know that you feel free to ask questions in the chat as we go through, but we'll also have some time for a Q&A at the very end for the 10 or 15 minutes or so. I'm going to put up another couple polls real quick. Thank you for answering that last one. There we go. And yeah, so we have quite a good turnout today. This is great. So there will be a recording. It's being recorded right now. I will be sending that out to everybody probably within the next 24 hours after the webinar is over. Okay. Great. Oh, we have a mix of people. Yeah, John, do you want to just introduce yourself while I'm doing the last poll here?
Yeah, well, thanks, Christy. Really glad to have everyone here. I'm John Harkness. I'm the CEO of Rewire Neuro. Yeah, we're really proud to show off kind of where Pipsqueak is now. It's really come a long way since, I think since some of you have started using it. We first developed Pipsqueak, way back in 2016 as part of some of my postdoctoral work, and really built from there to start integrating some of the AI capabilities that you're familiar with now, and turn this into a really robust platform that's capable of doing some very powerful analysis. Just in the last week, we released this brand new version, Pipsqueak 2.0, and it's great to see, I'm really excited to show you what we're capable of doing now. This has been a really big milestone for us. So we'll walk through some of those features. Yeah, feel free to ask questions as we go here. If I see them or if Christy sees them, I'll try to answer them kind of as we're going through things. And like Christy said, this will be available as a recording after. So, you don't need to jot notes furiously or anything like that. At the end, I will spend a couple minutes with general Q&A. So if you'd like to chime in then and turn on your camera and microphone, you're welcome to do so. I also have a link for you to book demos with one of us. If you would like to spend a little bit more time one-on-one, I'd be happy to walk through some of your images with you and potentially see what Pipsqueak can do for your analysis. So yeah, with that, Christy, I think we got our polls out of the way?
Yeah.
Great. Well, good. Yeah, so Pipsqueak has come quite a long way. We've got a few exciting things to talk about. And right off the top, I want to mention an integration that we just launched in the last 2 weeks with eLab Next, which is an electronic lab notebook. A really fantastic partnership. It's allowing eLab Next users and Pipsqueak users to port data back and forth between the two platforms, provides this really nice way to analyze images. And so I'll talk a little bit about that. of course, go into our new custom model generation engine in Pipsqueak, and then we'll spend a little time with Q&A. So yeah, just as a, as a background to Pipsqueak and its adoption in the research community, we first published the Pipsqueak method, which actually had more of an acronym when it was originally released back in 2016. And so our Slaker 2016 paper is one that some of you may have seen as a reference for our techniques, our methods. Since then, it's really grown. I think that paper has about 60 citations now, and including citations to our ID. And so if you're looking for others who have used Pipsqueak and what they're saying about it, those would be good ways to go. And of course, you can find a list of all those publications on our website. And so, yeah, eLab Next is a really exciting advancement for us. We're really thrilled with what eLab Next is providing, and I'm going to show you just a little bit of how that looks. And so what I've got here is my page on eLab Next, eLab Journal, loaded up where you can see one of the experiments that Grant and I were working on. And so this is a good place for me to import some images from that experiment. I can upload my protocols, upload my notes, and upload my results. And what's exciting about this is Pipsqueak is able to automatically export some of these images straight into the Pipsqueak interface to start the processing. And so we're going to head over there, and I'll show you how that looks. So great. Oops, here we are. So we're in the Pipsqueak interface here, and I just pulled up this nice image to remind me of some of the cool new features that I'm going to show you here in a second. But let's start off with just a brand new image upload. I'm already in a project here, and so let's see, I've got my project list, and I'm just going to go back right where I was into a project in Pipsqueak, and I'm going to show you how I can import images straight from eLab. So this is cool because all I had to do was bring in some of my credentials to automatically sync my projects in eLab up to Pipsqueak. And so I'm actually going straight into that project that I was just showing you. I can even pull up a specific set of images from there, and I'm just going to import this here. And this should take just about 10 seconds to bring, I think, around 10 images into our platform, and then we can start taking a look at how that integrates. While that's uploading, I'm going to come right back over here. So these are the images that we will be processing. And when we're done processing, we're actually going to see Pipsqueak upload, or have the potential to the option to upload both some of the ROIs or some of the detections that we created, and a comma separated value of our results. So this is a really good way to store, archive, and share all of those data. So great. Oh, all right, back here in Pipsqueak, our upload has been completed. So these are some of the images that we just pulled down from that project in eLab. So this is really cool. And so all I have to do now is select one of the detection models that I'd like to use to detect these cells and press detect. And so what we should see here is in just a second, all of these parvalbumin cells are detected, voila, just like that. And so, at this point, if you're familiar with using Pipsqueak, this is, just kind of your chance to go in and, do whatever else you want to annotate this image. You might want to come in and delete some of the detections that you don't like for whatever reason. Maybe those aren't cells that you want. And when we're done, we can just approve our cell detections. At this point, again, Pipsqueak users might be familiar with exporting our ROIs, which gives us the ability to see our comma separated values, to see that results folder file. Oops, and I think my connection's just getting a little laggy here. But what we can also do now is actually export those images into eLab. And so, while my computer's hanging here, I'm just going to go straight back over into eLab, and I'll show you what that looks like. So here I've got one of my files that I can open up. And if I go into, I'm just going to download that onto my computer. And now when I open that, we can see that I've got all of the ROIs that have been detected there. So that's really nice. This is something that's available to all the other lab members in my lab that have access to our eLab Next account. So if you have any questions on that, feel free to pop them into the chat. Otherwise, what I'd like to do is move on to some of our other Pipsqueak demos. And I'm just gonna open up my Pipsqueak here and one more time, and let's hop into this CFOS project that I've been working on now for a while. What I've got here is, potentially these are a number of images that I've already stained with a CFOS marker. I've imaged these, in this case, probably on my confocal microscope, because what I'm showing is both a single channel here and an associated merged image. And so, what I'm probably doing in a scenario like this is going through one of my channels one by one, because I would like to find all of the CFOS labels in this image. And so all I need to do this is to come into my model library and select the CFOS model, maybe adjust my sensitivity settings, and press detect. And when I do that, we should really quickly here see again, just like we saw last time, great, that there's a bunch of detections that popped up here. So, okay, I can kind of see what's going on here. Like I showed you before, I may want to go in and annotate some of these other cells that I would like it to detect. I have a couple options to maybe make the image a little bit easier for me to see. I can bump up the brightness, I can bump up the contrast. And what's really nice about this sort of feature in Pipsqueak is when I do that, I am not adjusting, I am not altering the quantification of the intensity that is ultimately going to be reported. These adjustments that I'm making in my image view settings are really just adjusting or improving my ability to see the image. And one of the new ways that we have to do that is here with these LUT options. So if you're familiar with using LUT coloration in lookup table coloration in ImageJ then this should be pretty familiar to you. This is sometimes a really nice way to get a little bit more information out of an image than is available in just a grayscale. So here I've imaged it with fire. You can do whatever you'd like. That's about the same. Great way to just visualize what you're seeing here. If I turn this off, we'll go back. And so, yeah, I think this particular model, this is an out-of-the-box model that's available in Pipsqueak, which is a really nice tool to have, is doing an okay job of detecting these CFOS marks. But maybe for my particular research style or my project, I'd actually like Pipsqueak to detect some of these more faint CFOS signals. And so to do that, what I'm going to do is I'm going to spend a little bit of time teaching Pipsqueak how to do that. Now, what's really cool in Pipsqueak 2.0 is that we made this process much, much easier. Again, if you're a previous Pipsqueak user, then you're probably familiar with this process taking quite a bit of time. We were asking for 2,000 annotations to be brought into Pipsqueak in order for us to train a model. And so, this was difficult for users to do. It was time intensive. Circling 2,000 cells is going to take you most of the afternoon. And then the training process, was a little bit slower than what we wanted to achieve. We were seeing training times take between 1 week and 2 weeks, primarily because we had people helping out with that process. So now in Pipsqueak 2.0, all I need to do is actually circle all of the cells in 5 of these images. And so, that becomes a little bit more possible for a user to do. And we've made some really big improvements for how a user can do that. So not only can I go in and delete some of those ROIs that I don't like, but I can also now use a stamp feature similar to what you might have experienced in Fiji. So if I highlight one of my cells and press Control V, all I have to do is click my, oops, I'm sorry, I gotta highlight it. Great, and when I press Control V, it's going to pop an ROI down wherever my mouse is pointing. So this is a much faster way to go through and use some of kind of the general morphologies that I've created from these, or general outlines I've created for these ROIs. So maybe I'll choose some large ROIs for large cells, some small ROIs for small cells. And basically, I'm going to make my way through this entire image to detect all of these cells. When I'm done, all I have to do is press approve, and I'm done with the image. One other thing I want to point out here just before we move on to custom model generation is I've got some really great abilities now. This is something a lot of our users have been asking for, to zoom and pan around my image. To zoom, I'm just using my zoom tools up here, or again, pressing Control and scrolling in on my image, and Control scroll out on my image, or Control click and drag around my image. So it's a little bit more like Google Maps now in terms of how you're moving around an image. I can pop right back out to my main frame, and I can turn my ROIs on and off, which I think is a really nice tool when I'm going through and determining whether my detections are how I'd like them to look. So great, I've done that. And as you can see, I've actually gone through and already annotated 5 images here. So we're already complete with all of the ROI annotations that I need to produce. The one caveat here is, of course, that I didn't finish annotating this entire image. And so I think that's one of the important things to think about as we were training AI detection models, is kind of this kind of the saying of garbage in, garbage out. And that really applies here too. So, if I am teaching, if I'm showing the machine learning engine bad examples of what I want it to detect, it is going to produce bad examples like that for me in the results. So the more time I spend really teaching the model what I want it to do, the better off it's going to be. And I think this is really similar to any sort of machine learning tool, AI tool that you're using out there, from any major company. You really need to spend some time giving it good annotations. Otherwise, it is going to return back something that you're not very happy with. So, in some of these images, I've actually spent a little bit more time going through and detecting everything exactly as I'd want it to be. Here's one. I'm going to just delete that. And then we will approve this image. Great. And now we're set to actually go over and customize this model, or develop a custom model. So another really exciting new feature here is we've cleaned up a lot of our interface. You may have already noticed that we're in dark mode here now in Pipsqueak. This is one of those features that I can switch from my Pipsqueak Pro options, in addition to my ROI shape and color. And I've got a lot of my navigation menu items here on vertical menus on the sides rather than being across the top. So a nice slick new interface, we think. And so I've actually got a couple things already started here. We're going to treat this a little bit like a baking show, so we'll pull the cake out here in the end. But what I'm going to do is I'm going to start a brand new model. Oops, I guess we want to call it what so we're just going to call it John's Demo CFOS model. Oh my goodness. Great. Oops. And we're going to associate that with a project. And so in this case, I want to associate it with the CFOS project that we're working in over here. And, I can just give it whatever description I'd want. A brand new feature here that I'm really excited to introduce is the ability to share the models that we train. And so this is very much similar to how you might experience sharing assets in Google Drive. But all I have to do here is essentially include the people that I'd like to share this with. So I'm going to share this with Christy Lawton. And all I need to know is, Christy Lawton. And all I need to know here is the email address that Christy uses. So I've got that one. I'm also going to share it with Grant. And so when this model finishes training, they're both going to get notification that it's finished training, and they're both going to see this model show up in their custom model library. So really nice little tool there. And we'll create this custom model project. And so when I did that, you see it pop up over here. This is basically an empty project for me now to add those images that we just started going through. So all I need to do here is is select the ones I want to use. Oh yeah, I like that one. I like that one. This is probably a good chance for me to be like, oh yeah, here's that image that I was messing around with. And this actually isn't one that I want to include. So, from one of my views, I can just unselect that one and let the model know that I don't want to include it. Maybe same with, these two that were empty. Great. And so I've got 5 images now that have the annotations that I need in them. And so I just press add, and that's going to pop them into my annotation library. And I think right now it's just hanging because of my internet connection. And so, while that's processing, I'm actually going to skip ahead a step. And so what we can see here is now I've actually already added all of the images that I need in my contour library. And so the next step that I'm going to do here is I'm going to start looking at those individual ROIs that I made during my analysis. And I'm going to actually draw the contour around these cells. And so this is a good chance for me to be very careful with exactly what I'm teaching our model. For example, this cell is a little bit hard for me to decipher, so I might actually skip that one and go ahead to this next one that has a little bit better of a example for me to see. So I can see my ROI around it. One thing that I'm seeing here is I actually don't really like how my ROI was placed. That's something that I'm going to need to be more careful with when I'm conducting my annotations. But now all I need to do is quickly just draw a little contour around the cell. And I want to be careful to make sure that I'm contouring exactly around the cell where I want it to, where the cell ends. I want to make sure, I'm going to delete this here. I'm going to make sure that my contour doesn't do something like this and include a bunch of background. And I'm going to make sure that my contour isn't too small and only including, the interior of the cell, not going around the edges of the cell. So in this case, I'll do that again. I'm going to draw my contour nice and carefully there. I like it. I press approve and I can move forward. And so what we'll see is that starts to count up. We've got 17 contours in there. And again, here I'm going to skip this that cell. Here's one that I already drawn. That's great. Another that I've drawn. Voila. And here's my last one. I like it. Okay. So now I've drawn all my contours. And as these contours are uploaded into my model library here, it's just, again, lagging here for a second as we look through these. I'm just going to keep adding some while it's counting. Great. So now we're a little ahead of where we need to be. So 21 out of 20 contours, that's great. A couple extra examples. All I need to do now is press train model. And so this comes up with just a little warning, talking about some of the things that we just mentioned: 5 images, 20 contours. And all I need to do is press train. And so once this happens, my model's been submitted. Now, if I go back to my model, my project library, I can see that one we were working on, which is titled here, step 2, is in progress. I've got a little training icon on that. So we actually trained one of these just last night in preparation for this webinar, and we trained it off of this exact data set that you're seeing here. And when I did that, it took actually about 6 hours to train it. This is the model here. And when it was finished training at about 1 a.m. this morning, I got this email that says the model name here is done. It's been deployed. And Grant and Christy both got that same email. So what's cool is if I go back over to my images, I'm going to choose another CFOS image here. We're back up into our analysis tab here on the right. So now I've got that CFOS model available to me in my library. And this is not the coolest image. So here's a cool image. So I'm just going to choose my brand new CFOS model. I'm gonna maybe adjust my settings to be specific for what I'm looking for, and I can press detect. And what we should see here is, yeah, wow, actually that's fantastic. It's detecting exactly what I want it to be showing me here on this image. We can go over maybe to another one. Let's do a little comparison. Let's take our initial CFOS model. I was running this up here, so we can detect with that. And okay, that looks pretty good. And we can go down and run the one that we've customized here. Bring my settings to where I want them to be. Run it again, and voila. All right, so this is a really nice way for me to go in and really fine tune the detection capabilities of Pipsqueak to be specific for me. This is a pretty small transfer that we've made going from CFOS to just an individualized CFOS model, but we've seen this technique work really well for users going from maybe from WFA over to something that's kind of similar but a little bit different, like microglia. So we now have a number of microglia models available. This is a process that can be iterative, or it can be one and done. If you've trained this and you come up with a result that works really well for you, then that's great. You can continue using this. Like I showed you, you can share these models with people in your lab. If I go back to my library here, I can say edit model, and so I can actually add in somebody here. So, oh, I forgot to add Grant to this model. So I'm just going to add him in. Awesome. And now Grant will see this model show up in his library as well. It's just going to think, because it likes to go slow when I'm trying to talk. All right, so yeah, we've got that. But yeah, let's say that maybe that I wasn't super satisfied with how this model was performing. There's actually a couple things that I can do to continue improving it. So this is an iterative process if we'd like it to be. So what I could do is I can either go into the model that we just trained, and this is now an opportunity for me to add more images to that model, add more contours to that model. These numbers that we have here, 5 images and 20 contours, is kind of what we consider to be a very minimum number of contours that are needed, examples that are needed to train these models. In actuality, there are hundreds of thousands of annotations that are required for these deep learning, machine learning models. And so we've got some really cool techniques to help this process along, bring that amount of work down so that you don't need to spend weeks or months doing these annotations. But we can add as many as you want. So if you got 40 images that you'd like to put in here and 100 contours that you want to show it, then that is going to be more information to show your model. And what's neat about that is I have really 2 options. I can either save over the model that I just trained, or maybe if I'd like to create some versions here, rather than saving over the one that I previously did, I'm actually just going to go recreate a model. Call it John's CFOS. John's CFOS like number 2, and I can start it from that exact same data set. And so this was this is that's a great way to kind of create versions of how our models are generated, especially if you're seeing just some differences in maybe how your images are coming off of the microscope. You changed staining protocols, you got new lasers in your confocal. Like things like this are going to change how images are being produced. And so it's easy to kind of tweak our detection models to be really specific for the types of images that we're looking at. So very easy to run that. And let's see. Oh yeah, and so there are a couple other really exciting features here in Pipsqueak that we've introduced in 2.0. One of the things that we've done is something that I was showing off here. Oops, take an image here. You know what? I think I'm just going to restart my Pipsqueak real fast. And so yeah, one of the things that we've produced here is hey Grant, would you pop in real fast and maybe take over for that for a sec? I think I just need to log back in.
Yeah, no problem. Are we on the right screen?
Yeah, I think he didn't show off the image analysis settings yet.
Cool. We're looking. Can we everyone see Pipsqueak?
Yeah.
Okay, great. Image analysis settings. Cool.
Although I don't know what John was just trying to show.
Yeah, well, exactly that, and then area selection.
Oh yeah, perfect. I can do that.
Yeah, I'm excited about the area selection.
Cool. So I'm actually just going to delete the ROIs off of this image to start with, because area selection has the nice feature of letting you pare down where the ROIs appear from inference. So let me start with, first you turn on area selection, and then it will let you choose the area you actually want to have on or have enabled in the image. And what we like to use the most for choosing the area is polygons, just because you can kind of draw it however you want to. So let's say in this image, I actually want, kind of just this general center area. I don't really care about much else in there. So we'll call that as our polygon. Not as good at drawing them as John with the fancy spiral. So then you hit save. Or I might have done that in the opposite order. There we go. It's a little hard to see on this image since I have a lookup table on, but on the outside of it right here, everything has gone to black. So now we only have selected that interior area that I had drawn with that polygon. So now if we come up here and run the CFOS model on this image, we should only get ROIs that are inside of that selected area. Yep, there we go. So all the ROIs are within the bounds of your area selection. So if we turn that off real quick, you can see that it didn't select any of the CFOS cells that were outside of that particular selection. And you can turn that back on real quick and it will go right back to that same selection.
This is one of those tools that we've seen really effective for analysis of like hippocampal regions. If you're going in and just looking for a small area within your image, this makes that possible. Or in a situation where you have like a either an artifact, a rip, or I don't know, a hair going across your sample, or like a white matter track going across your sample, something that you don't want to analyze. This is a great way to remove that.
Yeah, definitely. And one of the nice things you can also do with it is, let's show the whole image again real quick. You don't have to select just one area. You can also select, like, say you have two areas in particular you're interested in. You can select those, and then you just have those two spots. So there's not really a limit to how many like sub-regions in the image you can keep. So if it was like a pretty complex image, it could be like, I just want these particular areas, and then you could run inference and just get those spots you're looking for. Do we also want to look at background subtraction, or?
Yeah, let's show this off real fast. So background subtraction is I think a pretty important part of analyzing signal intensity. So if you're quantifying the intensity of your fluorescent mark, it's generally accepted practice, I think, to subtract out background staining. And one of the ways that Pipsqueak is able to do that has been around for quite a long time. We've published this in our 2016 paper. It's using a couple different methods that are all widely used or widely available in ImageJ. And the first is just a rolling ball subtraction. A rolling ball smoothing. This is an algorithm that's been published since like the 70s, I think, that's basically allowing us to smooth out hotspots in our image. But what's important about rolling ball is to determine, the average size or the general size of the marks that we're trying to see. If our rolling ball is too large or too small, we'll see that it's smoothing over things that we don't want it to. And so we have some specific literature on how to size that on our website, or you can find that paper, I think, linked from our website. So you can try that out. But we also give you the ability to measure general background fluorescence or background intensity here. And that's done from these 20 some odd ROIs that we have spaced around the image. One of the other ways that I think it's fairly common for people to measure background staining is to, just draw 3 regions around an image. You can do that here in Pipsqueak also. These the standardized grid is used as really just a method to use the exact same locations from image to image. That's maybe one way to standardize what you're doing, but you can draw them in if you'd like. And essentially
also put them wherever.
Yeah,
right.
Yeah. Move them around, which is, I think an important part of this, to take measurements from around an image. And what that's going to do is it's actually going to measure that staining, and it's going to take just a section of our intensity spectrum. So we're going to ignore the brightest third of pixels that we measure, which are probably signal. We're going to ignore the dimmest third of pixels, which could be, areas of the image without staining, maybe areas of the image without tissue, if it's a rip, and come up with a numeric value in the middle that we subtract from our intensity score. So something you can turn on, turn off if it's not matching with your protocol. It's something you don't need to use, but we think that that's a pretty handy feature to have in here. And then Grant, one of the things that I'd like to just show here very briefly, because it's not the intent of this webinar, is our co-localization capability. And so, yeah, you might have to select some specific images to do that here, but this is I think one of the things that Pipsqueak is really known for. It's something that we've been doing since the very early days of Pipsqueak in ImageJ and Fiji, because a lot of confocal staining protocols will include staining of several different marks. In this case, we've stained for parvalbumin, CFOS, and perinodal nets. In this case, stained with a Wisteria floribunda agglutinin stain, WFA. And so, here we've got those three channels and we've got a merged image. And so what Grant has done is he's just chosen the specific detection models that he wants to use on each one of those channels, indicated them, and then indicated our merged colorized image up there. And so he's going to detect them. That's going to run each one of those detections simultaneously, which is pretty fantastic because, voila, you've done 3 images at once. And it gives you the chance to click in on them, maybe, change any of the ROIs that you would want. You can, of course, apply a custom detection model like we just trained here as well. And eventually we'll take these detections and co-localize them. So everything that you'd be doing on a single channel, but now you can compare those localizations on your merge. So he'll press run co-localize, and voila, we've got 5 cells there that co-localize. What's really great in this version of Pipsqueak is you can now go in and get a little bit more information about what else was detected here. So one of the things that I think is fairly common to do when co-localizing is to look at your merged image and look for your merging colors to tell you, oh, well, purple is where I'm going to see all 3 of these marks show up. I think one of the problems with that method is that our eyes can trick us into seeing things that aren't there. And so this tool now gives you the ability to see maybe why Pipsqueak didn't detect something that you think is co-localized. It also allows you to maybe determine if that was an error on Pipsqueak's part, and you can go back to those individual channels and correct maybe where that particular ROI is located so that your co-localization does show up. But
yeah, like you were to look at one in particular, like this one could be, a potential.
Yeah, like that looks like that parv is just outside of the WFA box that's measured. So, that'd be a good chance to go back to your WFA individually and decide if that's actually the cell that you're trying to detect. So yeah, pretty powerful way to co-localize. And then of course, these ROIs can be exported as well and opened up in a comma separated file, just as you've seen before. I don't know if yeah, there you go. So he's opening it up here, and real quick, just what I'll point out with these results, again, a huge improvement over maybe how Pipsqueak has operated in the past is we're now combining all of these results files into one. So rather than having to open, like 24 results files for a triple stained image set, we're going to see all those results as one. We'll see where it co-localizes as one, so that you can start taking these results straight.
Excel to open this in right now, so this is opening in a code editor.
True development environment, but yeah, if you have Excel on your computer, your CSV will open up in columns just right there.
But what to look out for is we have the 5 co-localizations mapped up for parvalbumin right here. And then if you scroll down to the WFA section, you can see them right here. And then the CFOS ones are right here. So you can use those mappings to decide what did co-localize, and then you have all the other ones that didn't co-localize also.
So yeah, this is a large part of what we wanted to show you today. And Grant, if you want to hand the screen back over, I'll take us on and show us some of the ways that people can be using Pipsqueak. I will share my screen. Excellent. Yeah, so thank you, Grant. The Pipsqueak is something that you can download from our website right now. I'll show you a place to do that here in a second. But we've actually got some brand new options for how you can access Pipsqueak in addition to the month trial license that's available to you. We have our standard single user license that has been available in Pipsqueak for a very long time. And recently, we also introduced student monthly pricing, which is intended to make this accessible for students in labs that might not be able to purchase a Pipsqueak license right away. So month to month, if you want to just use this for a month or 2 months at a time, that's great. It will renew for as long as you want to use it, but it gives you the ability to train custom detection models, all the processing that you'd want to do, and something that you can control on your time. So if you need it tonight because you have a deadline tomorrow, then this is something that's available to you right now. We also have a brand new license that we're calling a lab license. And essentially, this gets rid of some of the restrictions that our single user license has had in the past. So now a lab license is available to as many users in your lab as you'd like to include. This is an unlimited number of users. It also allows a lab to have some admin controls and some sync and share capabilities, both features that are coming online here very shortly. So this is, primarily something that's going to be used in an environment with or in a highly collaborative environment where users are sending images back and forth. It's going to allow me to sync my data sets from my lab computer to my home computer or the microscope computer to whatever, my laptop, so that we're not having to carry around, terabyte hard drives or whatever anymore. We think this is a really great tool, kind of bringing some of the features that, or, the ease of document sharing and like Google Drive into this Pipsqueak environment. Of course, this is also allowing you to train as many custom models and share those custom models with other members in your lab and process as many images as you'd like. These licensors are also available at a number of different lengths. So this is something that we can put together with you on an individual basis. And so we've got Pipsqueak available in a 1-month trial. If you would like to download that, you can of course find it on our website, but if you've got your phone handy, you can scan this QR code or just take this URL, and it's going to take you into a spot here where you can find that trial. And I've also got the link here to book a meeting with me. I would be more than happy to hop on a 30-minute call with you, start taking a look at some of your images and see how Pipsqueak could work for you, and help you get going with it. So yeah, that's pretty much all that I wanted to cover, other than to point out our fantastic blog written by Dr. Christy Lawton. This is a really good chance to just kind of see a little bit of what's going behind the scenes. Christy talks a little bit about our machine learning, how it works, best uses, best practices with machine learning. So I think it's kind of a neat spot to stay on top of what's going on. And of course, we are just getting ready to go to the Society for Neuroscience conference in sunny San Diego, which is nice because it just started drizzling here in Portland today. Next month in November. So if you're going to be at SFN, come by our booth, grab some swag and check out what we're doing. Or Cell Bio. This is the first time we've gone to Cell Bio in DC in December at the end of the year here. So come by and say hi to us there as well. And yeah, I am more than happy to answer any questions that anyone has. And like I said, if you want to reach out to me after, my email is here, or that QR link that you can find in the recording. So thank you so much for coming by. I think while you're answering questions, Christy has another poll that she's going to put up, and so that will just kind of be going on in the background as we talk here.
Yeah, and I'll also put our emails in the chat here in a few minutes. But yeah, if anyone wants to, you can unmute to ask a question directly or type it in the chat. Either way. John, are you done? Are we ready for Q&A now?
Yeah. Anytime, happy to answer any questions. Yeah, I think this is kind of one of those big evolutions in the platform here. So it's pretty cool to see, some of our users come along from the early days of Pipsqueak, struggling through using it and like ImageJ and Fiji, and making, these more powerful features available. We certainly have an aggressive roadmap ahead of us. We've worked out a number of features here that will be coming online within the next 3 to 6 months that are really going to improve the ability to sync, share, collaborate, and process these images. This is also a great time to hear back from some of our longtime users if there are things that you've found important in your workflow. We're really happy to hear about ways that we might be able to improve Pipsqueak for you more specifically. So question here, reproducing, how would you recommend formally reporting a custom model? Excellent question. Yeah, thanks for asking that. So we have taken a philosophical stance in terms of our custom model development that the models that are available to you out of the box with Pipsqueak are things that are not going to change. They might be versioned, and if they're versioned, they will come with new names. So, we were looking at that CFOS model a lot today. That model is going to stay the same. And if you'd like to, if you'd like to use a different Pipsqueak, or I'm sorry, a different CFOS model, that would be developed to you, or developed version-wise. Citing those models is possible by citing our ID number that I had there on the front. It's also available on our website. And what I would recommend doing is saying specifically which model you used. In the future here, we're also allowing users to share models that they've generated with the community. And so I think this is a really good way to take ownership over your methods. So for example, if Grant trained a CFOS model, he might publish it as Grant Wade et al., and make that available to other users in the community, at his discretion. Yeah, great question. Thank you.
Hey, John, I had a quick question.
Yeah.
So I was playing with the version 1 and version
Trevor,
2. Yeah. And the output gives you the data for every region that you create. Is there any plan to move into more of a summary analysis and just give you results on number and average and intensity or things like that? Because if you're working with 2,000 images and you get 60 ROIs per, it's just going to be quite a cluster.
That's a great question, and that's something that I think you can really start to approach 2 ways. The first is our integration with eLab Next. I think provides a lot of that capability to some extent. It makes it possible for you to upload those data into eLab Next, and they've got some data analysis tools in there. We are also working on some capability to actually export your data either as a project into a database, so that you might be able to work with it there, or to use some of our kind of pre-curated database functions to pull out things like mean and average, or mean and standard deviation, or whatever it is. Part of the strategy with that is coming up with ways for you to provide grouping variables, and so that's something that you're also going to see in your project tab. So you might be able to create a folder and say, the 10 images in this folder are, group A, treatment group, and the 10 images in this folder are the control group. So you'll start to see the ability to grab those descriptive statistics a little bit easier. Yeah, great question, Trevor, and great to see you.
Thanks, John.
Well, yeah, thanks again, everybody. And like I said, we're here to chat with you. If you have anything else that comes to mind. So thanks so much, and look forward to seeing you at SFN or at CellBio.